And our final speaker for the day is Dr. Ankit Nagar, who is actually our most recent trainee to join the lab. He's a postdoctoral fellow in our modeling and simulation program, working on some of our projects right now in Alzheimer's disease. So, Ankit.
Thank you for the introduction, Sarah. Good evening, everyone. I would like to walk you through my research on model-informed preclinical study design for an approved anti-amyloid immunotherapy in one of the newer mice models for Alzheimer's disease, which is APP-SAA mice. We are using an approved immunotherapy to validate this mice model so that when we test newer drug candidates in this model, we can attribute the success or failure of those future trials to the drug candidate itself rather than the model, and avoid or minimize the risk of any false negatives. Amyloid is a peptide species found in the brain, and two particular amyloid-beta monomers, which are of the size of 40 and 42 nucleic acids, have a larger tendency to aggregate in the brain and lead to bigger size species like oligomers, protofibrils, and subsequently plaques. So these plaques are insoluble sticky substances that deposit into the brain and are central to neurodegeneration and Alzheimer's disease. There are multiple immunotherapies which have targeted different amyloid beta species in this aggregation cascade, and several of them have failed to actually successfully translate in the clinical stage. And the primary reason for their failure is that these mice models, which were used to test these immunotherapies, overexpress the amyloid precursor protein as well as these A-beta species in the brain, which is several folds higher than the physiologic levels. And therefore, you see an inflated treatment effect in the preclinical stage, but does not translate successfully in a later stage of clinical development. So you see these failures occurred in phase 3, all of them. And therefore, there is a need to come up with newer mice models which produce these amyloid beta species at physiologic levels, and therefore we can observe treatment effects which are closer to true treatment effects and improve the translational relevance. Coming to the APP-SAA knock-in mice model, so this model is relatively new and it contains 3 different humanized mutations which allow us to increase the production of A-beta monomers and protofibrils, as well as shift the aggregation towards more A-beta 42 driven. So this is closer to physiologic levels compared to prior reported mice models, and gives us a closer chance of detecting a treatment effect closer to a true effect. And therefore the question is, how do we design a chronic immunotherapy PK study using this mice model? We are using Lecanemab as our approved immunotherapy, which is known to target large-sized oligomers and protofibrils, which are soluble aggregates in this cascade. The question or the decision we are trying to make here is what dose levels of Lecanemab do we test in this trial, and what should be the baseline age of mice to start the trial with in order to observe a 30% or more reduction in plague burden in these mice from baseline for a trial duration of 17 weeks. So it's very specific, and we built a semi-mechanistic QSP model to answer this question. This is the overall workflow. We had already published PBPK QSP model from Gertz et al., and we adapted this model to focus on Lecanemab relevant biology. This human QSP model was then qualified on the clinical data that was available from the clinical study for Lecanemab. This qualified human QSP model was then reparameterized to be a mouse model, and we had the PK data, in-house PK data from pilot PK study to inform the PK parameters. After qualifying our mouse model, we conducted simulations to see what dose and baseline age levels allow us to capture the dose response for this mouse model. Coming to the human QSP mouse model structure, so this is one of the simplest slides. And you'll see that I have 2 compartments, plasma and brain, and they are separated by a blood-brain barrier. The entire aggregation cascade starting from amyloid precursor protein all the way to formation of plaques is shown here, happens in the brain compartment. And there are protofibrils and large-sized oligomers, which are targeted by Lecanemab, and they are in equilibrium with the free states. We used this UN-QSP model and then simulated and overlaid on observed clinical data. The left panel shows y-axis with PET SUVR, which is a surrogate for plaque burden. And you see that the model is able to adequately capture the longitudinal trend in observed data at a dose level of 10 mg per kg, Q2 weeks and Q4 weeks. We also have a secondary endpoint which represents that A-beta-42 species are getting cleared from brain into the plasma, and there is a rising concentration relative to A-beta-40, and the trend is captured reasonably well here as well. Subsequently, after qualifying the human model, we scaled the human model by applying these multiplicative factors on certain rate constants in this aggregation cascade, which are known to be modified because of the presence of these three mutations, Swedish, ostrich, and Arctic. And these multiplicative factors were informed by the observed plaque trajectory data we had collected in-house. Y-axis here is plaque density and x-axis is age of mice. You see that in the observed data that plaque burden is gradually increasing from about 4 months of age till up to reaching saturation beyond 15 months or so. And our model was able to adequately describe the trajectory of the plaque burden over mice age. Based on our in-house PK data, it described a one compartment model adequately described that data at a 3 milligrams per kg intraperitoneal administration. And these are the parameter estimates from the one compartment PK model. And this PK model was then incorporated in our QSP model, which I showed on the previous slide. And the entire qualified animal QSP model was then used to simulate our primary endpoint, which is % reduction in plaques from baseline at a 17 weeks trial duration. We simulated 3 different dose levels ranging from 3 milligrams per kilogram to 30 milligrams per kilogram, and along with the control arm. And you observe that there is a wide range of dose response, and the range is wider when you start at a younger age of mice compared to a much older mice to start the trial with, where you see a much compressed dose response towards the end of the trial. And therefore, it is favorable to start the trial as early as 4 to 6 months window, where you have an onset of plaque generation in the brain, and the treatment effect is wide enough to capture across these dose levels. That leads me to the conclusion, which is the final study design itself, where we have 5 different arms. The genotyped mice is in 4 of these arms, 3 of them being the dose levels I showed the results for, and 1 being the vehicle arm. We also have a wild-type mice arm, and we have equal proportion of males and females across all the arms. And we are planning to start the trial at a baseline age of 6 months. And we would like to collect, with regard to endpoints, protofibrils and plaque density in the brain, along with conducting behavioral tests to inform the cognitive behavior of these mice. We also plan to collect PK samples at 4 and 8 weeks, pre and post at the steady state. Leading me to the acknowledgements, I would like to thank my PI, Dr. Sarah Quinney, as well as special thanks to Dr. Paul Terito, who has helped me provide the feedback and improve, and along with other postdoctoral committee members, as well as division chief and my lab members. Thank you so much.